Once he's dead, he's taken off the cross and wrapped in cloth and taken to a borrowed tomb.
And because of the contact with the body of Jesus, that transforms and leaves a lasting mark on the cloth.
In 2015, inside an ultra-clean genetics lab at the University of Padua, Professor Gianni Barcaccia and his team cracked open something never supposed to be found. A secret DNA code music hidden inside the Shroud of Turin. The cloth millions believe wrapped the body of Jesus Christ after the crucifixion. For centuries, the debate was simple. Either the shroud was a medieval fake or it was real. The DNA was supposed to settle it. European DNA meant forgery. Middle Eastern DNA meant authenticity. But when Baracca's computers finished decoding millions of nucleotide sequences and the final genetic map hit the screen, his team just stood there silent. Because the secret code didn't point to one person, one country, or one continent. It pointed everywhere. The DNA mapped into that cloth revealed a journey so vast and precise that it couldn't have been planted, couldn't have been forged, and couldn't be explained by any theory ever proposed. Here's what they found. The face no one was supposed to see. To understand why that DNA shattered every existing theory, we need to start with the night the shroud first revealed what it was hiding. May 28, 1898. Tin, Italy. A lawyer and amateur photographer named Sacandopia was granted rare permission by King Umberto I to photograph the relic during a public exhibition. Photography was brutal work back then, slow, technical, unforgiving. Pia hauled a camera nearly the size of a suitcase onto scaffolding inside the cathedral. Using magnesium flashes to light the dark interior, he exposed 2 large glass plates. Late that night, alone in his darkroom, lit only by the dim glow of a red safety lamp, Pia lowered one plate into the developing chemicals. As the image materialized, his hands started shaking. He nearly dropped the glass. What was forming before his eyes wasn't supposed to exist on the photographic negative, where light should become dark and dark should become light. A sharp, high-contrast face appeared. Deep-set eyes gently closed. A broken nose, a mustache, and forked beard, bruising along the right cheek. For the first time in history, the figure hidden inside the linen wasn't a faint shadow. It was clearly a man, calm, dignified, yet marked by extreme suffering. Now here's the catch. There's a sense of urgency about moving Jesus' body from the execution site to the burial site. Any normal image behaves predictably as a negative. Light turns dark, dark turns light. And the result looks distorted, unnatural. But the shroud broke every rule. Photographed as a negative, it appeared as a true photograph of a real human being. What the naked eye sees on the cloth is already a negative, who, in the Middle Ages, understood photography 800 years before the camera existed, who could create a flawless negative image with no way to see, test, or confirm the result. No one. The human eye cannot view the world in negative, let alone reproduce it with precise control over light and shadow. No artist, no scientist, no scholar alive at the time possessed the knowledge to even conceive of such a thing, let alone execute it flawlessly on a piece of linen. This was the first crack in the wall of skepticism. The shroud didn't behave like a painting. It behaved like a photographic plate, capturing a single instant in time. And that realization sent a tremor through every discipline that would later touch this cloth. If this is already pulling you in, hit subscribe, because the DNA was only half the secret. The other half is something nobody saw coming. But that photograph, that was just the warm-up. The real bombshell was hiding in the dust. And when they finally decoded it, even the skeptics went silent. The DNA that shouldn't exist. In the 21st century, speculation finally gave way to something much harder to argue with. Genetics, high-energy physics, spectroscopy, forensic molecular analysis. Scientists stopped treating the shroud as a holy relic and started treating it like a crime scene, a biological data vault, quietly preserving 2,000 years of history. In 2015, Professor Barcaccia's team of geneticists and biologists was granted unprecedented access to the relic. Their goal wasn't to find the DNA of God. Science has no model for what such a trace should look like. They wanted to reconstruct the life story of the cloth itself, where it had traveled, who had touched it, whose hands had held it across the centuries. That leaves us with a very real possibility we could be talking about a first-century cloth. Using sterile microvacuum devices equipped with ultrafine filters, they collected microscopic dust, pollen, and organic fragments from deep between the warp and weft music threads. Spaces where ancient material can remain sealed for thousands of years. And get this, they focus specifically on mitochondrial DNA. And for a critical reason, unlike nuclear DNA, mitochondrial DNA exists in hundreds of copies per cell. It's passed down exclusively through the maternal line, and it survives far longer in ancient material. It's essentially a biological GPS, a powerful, reliable marker of geographic origin, population movement, and human migration. If any genetic information had survived inside those fibers across 2 millennia, this was the method that would find it. For weeks, computers ran nonstop, decoding millions of nucleotide sequences and comparing them against global genomic databases representing populations from around the world. Then the final maps appeared on screen. Haplogroups, ancestral lineages, geographic origins. The lab went quiet. Nobody spoke. Baraka's team was staring at data that defied every prediction they had made. This was not the genetic signature of a single individual. It was something far more complex, something no one had anticipated. The findings published in the highly respected journal Scientific Reports detonated across the scientific community. Here's what they expected. If the shroud were a medieval forgery created in France, European DNA should have overwhelmingly dominated. If it were an authentic relic from Jerusalem, Middle Eastern DNA should have dominated. Neither happened. First hit, the Middle East. Haplogroups commonly associated with the Druze, a tight-knit, historically isolated community in Israel, Jordan, Lebanon, and Syria, whose genetics have barely changed in millennia. That alone was significant, a powerful marker of geographic origin, providing strong evidence of a direct Middle Eastern connection dating back centuries. But then came the second hit, Western Europe, haplogroups, U5b and H1 through H3, consistent with centuries of European handling. The Poor Clare nuns who repaired it, the House of Savoy who owned it, countless pilgrims who venerated it across the centuries. Okay, that tracked. You could explain that. But here's where it gets wild. Third hit, North and East Africa. Haplogroup L3 pointing toward Egypt and Ethiopia. That was unexpected, deeply fascinating. It suggested direct contact with some of the earliest Christian communities on the African continent. The room shifted. Fourth hit, South Asia. Flow groups M39, M56, and R8. Genetic markers typical of the Indian subcontinent. Now the team was staring at each other. And then the fifth hit landed, the one nobody could process. East Asia, haplogroups D4 and G2A commonly associated with China, India, Africa, Europe, the Middle East, all encoded into a single ancient cloth. Let that land for a second. But how? And if a forger didn't put it there, who did? The answer was hiding in an ancient route that most scholars had already written off as myth. A world written on cloth. Now pay close attention to this. No medieval craftsman working in a French abbey around 1350 could have collected genetic material from China, India, and East Africa. If you mess with us, if you mess with the Roman Empire, this is what will happen to you. You will die, and you will die horribly and publicly. Globalization didn't exist. Long-distance travel occurred, but not on a scale vast enough to leave such widespread genetic signatures on one piece of fabric. Nor could such a person have intentionally planted music, those traces, in a way that would fool genetic scientists 600 years later. The DNA found on the shroud is not random contamination. It is a collective biological memory. But here's the deal. When Barcia mapped the data geographically, the mystery deepened even further. The distribution of genetic markers aligned with remarkable precision along the Mendelian route. The ancient path scholars had long dismissed as legend. According to early Byzantine, Syrian, and Arabic sources, the shroud was folded so only the face was visible, a form called the tetradiplon, and displayed inside a special frame. This relic didn't stay in one place, and it certainly didn't begin in Europe. Its journey started in Jerusalem. From there it moved to Edessa in the 2nd Music century, where the shroud remained hidden within the city walls for centuries. And this is where it gets fascinating. Music. Edessa sat at one of the most important crossroads of the ancient world, the Great Silk Road. Caravans from China loaded with silk, merchants from India carrying spices, Persian diplomats, Arabian traders, pilgrims from every corner of the known world. And many of them came to venerate the image believed to protect Edessa. They stood close. They kissed its casing. They touched the cloth. And with every encounter, microscopic traces, skin cells, hair fragments, droplets of sweat settled onto the fabric, layer by layer, century after century. The DNA of the entire ancient world accumulating on its surface like invisible dust. The journey didn't end there. From Edessa to Constantinople in 944 A, the greatest megacity of the ancient world, where people of every race and culture converged. Then in 1204, during the Fourth Crusade, the city was violently looted. The shroud disappeared. It resurfaced in Athens around 1205, passed through French knights, and finally appeared in France around 1353 in the possession of Knight Jeffa Dessari. And it is here that the genetic music evidence delivers a devastating blow to the medieval forgery theory. The DNA on the shroud is not random noise. It is a geographic fingerprint that no forger could have manufactured. Dust from sandals, robes, and hands, left behind by countless people who stood before this face over many centuries. Music, a biological trail stretching across 2,000 years of human contact. Something of that nature, in medieval times, or even in the 1st century, just couldn't exist. But the genetics were only half the secret code. The other half was written in plants, and it pointed straight to a crime scene. Pollen, blood, and a crown of thorns. Professor Abinoam Danan, a renowned Israeli botanist from the Hebrew University of Jerusalem, and Swiss forensic scientist Max Frey, independently examined pollen grains trapped deep within the linen fibers. When Danan saw the complete results, he knew the implications were enormous. 58 different plant species identified, cataloged, mapped, 17 native to Europe, expected given centuries of European handling. But the majority came from elsewhere. The Middle East, Turkey, the Anatolian steppe, perfectly matching the ancient route through Edessa and Constantinople. And get this, scientists identified plants that grow nowhere else on Earth, only within a narrow corridor between Jerusalem and Jericho. One species dominated Gundelia toi, a thorny desert thistle. Its pollen made up nearly half of all samples, concentrated heavily around the head and shoulders. Now stop and think about that. Why would thorn pollen saturate a burial cloth? The crown of thorns, Gundelia torn 40, with its long needle-like spines, is exactly the plant Roman soldiers could have twisted into a mock crown. It blooms near Jerusalem in early spring, precisely during Passover. A second botanical clue came from Zyophyllum domosum, a plant endemic to a very restricted region, growing only in the Judean desert and parts of the Sinai Peninsula. Its pollen was also found in significant quantities. Here's the deal. No medieval forger working in France could have obtained pollen from plants native exclusively to Israel, let alone applied it invisibly at a microscopic level across a piece of cloth. A forger would have used paint. Pollen cannot be painted. It functions like an invisible seal from a crime scene, a geographic fingerprint that cannot be faked. And the blood told its own brutal story. For years, skeptics argued that the reddish stains were nothing more than pigments, ochre, cinnabar, or tempera mixed with gelatin. That claim collapsed in 2017. A team led by Professor Julio Fani at the University of Padua, working alongside physicians from a hospital in Trieste, examined the stains using transmission electron microscopy and Raman spectroscopy. At the nanoscale, they saw not pigment, blood, human blood. Blood type AB, one of the rarest blood groups in the world. This same blood type appears repeatedly on ancient Christian relics, including the Sudarium of Odo, a cloth tradition, says covered Christ's face at burial. But here's what stopped them cold. This was not the blood of a healthy person. Within it, scientists detected nanoparticles of creatinine and ferritin bound to hemoglobin. Such extreme concentrations occur under only one set of conditions: catastrophic fatal trauma, prolonged torture combined with dehydration and massive muscle destruction. It takes roughly 6 hours for Jesus to die. Once he's dead, he's taken off the cross and wrapped in cloth. When muscle tissue is repeatedly damaged, a process known as rhabdomyolysis, creatinine floods the bloodstream in enormous amounts, and the blood preserved on the cloth recorded exactly that. This is not symbolism. It is a biochemical signature of agony. The man wrapped in this cloth was beaten to a physical music state already incompatible with life before crucifixion even began. Evidence of over 100 blows consistent with Roman scourging using the flagrum, leather whips embedded with lead weights, and now this. Because this detail still haunts researchers, the blood stayed red, which should be impossible for ancient blood. Normally old, blood darkens to brown or black within weeks. But scientific analysis revealed unusually high levels of bilirubin, a substance released by the liver during extreme stress and severe trauma. Bilirubin preserves the red color of blood. For centuries, the blood of a tortured man stays red. Not mysticism, biochemistry. Under unbearable stress, a painter can imitate the appearance of a wound. No human hand can reproduce the biochemical signature of extreme trauma, organ failure, and hypovolemic shock. Every molecule pointed to the same place and the same event. But what about the one test that was supposed to prove it was all a fake? The test that got it wrong. In 1988, 3 prestigious laboratories, Oxford, Zurich, and Arizona, Carbon-14 dated the fabric to between 1260 and 1390 AD medieval, a forgery. Case closed. The world accepted the verdict. Even the Catholic Church stepped back, referring to the shroud as an icon rather than a true relic. But hold on, because science doesn't stand still. 3 decades later, researchers discovered where the critical mistake had been made. And the problem wasn't the technology. It was where they took the sample. In 1988, a tiny piece of fabric, no larger than a postage stamp, was cut from the very edge of the shroud. That corner had been handled countless times, absorbing sweat, skin oils, candle wax, and bacteria. Then, chemist Raymond Rogers of Los Alamos National Laboratory found something devastating. That corner had been expertly repaired during the Middle Ages. The Poor Clare nuns reinforced the damaged edge by weaving new cotton threads into the original fabric, dyed with alizarin and sealed with gum arabic to match the aged linen. Here's the kicker. The main body of the shroud contains no cotton at all. It is made entirely of linen. The laboratories didn't date the shroud. They dated the patch. Now pay attention to what happened next. In 2022, physicist Liberato Darrow at the Institute of Crystallography and Bari introduced a radically different approach: wide-angle X-ray scattering or wax. Instead of analyzing contaminants, this method examines the aging of the linen's cellulose itself at the atomic level. Over time, linen naturally degrades. The polymer chains of cellulose slowly break apart under exposure to radiation, humidity, and temperature. This process functions as the material's internal clock. Darrow compared samples from the shroud with fabrics of known age, from Egyptian mummy wrappings dating back to 3000 BC to medieval textiles. When the aging curve appeared on his monitor, Darrow called his colleagues over immediately. His disciples describe him as his face glowing, his clothes dazzling white. The shroud's molecular structure didn't match medieval fabrics at all. It was far older. It matched linen recovered from the fortress of Masada in Israel, dated between 55 and 74 AD, 1st century, the time of Christ. In 1988, science dismissed the miracle. In 2022, it brought it back. But one mystery still towers above all the others, and modern physics cannot explain it: the impossible image. No brushed